introduce our first speaker, Dr. Tanya Mayadas from Harvard Medical School. She will discuss leukocyte recruitment in the context of immune complex deposition.
I would like to first start with some acknowledgements. I would like to acknowledge my very talented graduate student, who's Florencia Rossetti. Let me see if I can see her. Florencia Rossetti, who just recently graduated, as well as my former postdoc now, Taki Suboi, who's now has his own group at Nagoya University. So the kidney actually filters about 40 gallons of blood every day because it basically filters the entire blood volume every 45 minutes. So in order to do this, it has actually developed a very sophisticated and ultrastructurally complex microvasculature. So the filtering unit of the kidney is actually the nephron, and we have about 1 million of these per kidney. And the nephron is composed of a glomerulus and a renal tubule. The glomerulus is fed by an afferent arteriole, which then subdivides into capillaries, which then merge and exit as the efferent arteriole. And then we have the peritubular capillaries, which are important for reabsorption of filtered water, salt, and solutes. Now today I will really be focusing on the glomerulus because diseases of the glomerulus constitute one of the most important problems in nephrology. So the glomerulus is composed of capillaries, as I just mentioned, and those capillaries are basically lined by a fenestrated endothelium. And then these endothelial cells then sit in a glomerular basement membrane. And on the other side of the basement membrane is the visceral epithelium, which have their foot processes on the basement membrane, and is very important in the size and charge selectivity for the filtration function of the kidney. You then have the parietal epithelium, which is in the Bowman's capsule, and they line the urinary space, where the plasma filtrate initially collects. So immune mechanisms underlie most forms of the glomerular disorders, and glomerular deposits of IgG, often with complement components, are found in the majority of individuals with glomerulonephritis. Now, the mechanisms of immune complexes formation really differs in terms of in situ. You can have in situ immune complex deposition, where antibodies recognize intrinsic tissue antigens in the glomerulus, such as the NC1 domain of type 4 collagen antigen, which is part of the glomerular basement membrane, and that can lead to anti-GBM nephritis, also Goodpasture syndrome. You can have antibodies that are generated to the PLA2R antigen, which is actually on the other side of the basement membrane, and that's in the podocyte. That leads to membranous glomerulopathy. You have antibodies that can bind to mesangial antigens. You also have antibodies that can react to planted antigens, which deposit in the capillary wall. In terms of, for example, circulating immune complexes that are formed in systemic diseases, example would be lupus. Those antigen-antibody complexes can also deposit in the glomerulus, and I wouldn't talk about these last two in the interest of time. So whether antibody actually generates an inflammatory response depends on where the antibody is located. If it's circulating immune complex, one important concept of the fenestrae in the glomerulus is that they're open fenestrae, so that the deposited immune complexes have access to circulating blood elements and leukocytes. So in the case, for example, in lupus, where you have circulating immune complexes deposited, you have subendothelial deposition, as well as in anti-GBM nephritis, where the antibody recognizes, as I mentioned, collagen type 4, you have now a very robust inflammatory response to those immune complexes. When you have, on the other hand, immune complexes that form outside of the basement membrane, such as in the podocyte, such as in membranous nephropathy, you do not have a inflammatory response. So glomerular inflammatory response usually entails hypercellularity, where you have leukocyte infiltration. You result in endothelial mesangial cell proliferation. You also have epithelial cells. This is more the parietal epithelial cell proliferation, which then gives, in combination with leukocytes, the formation of crescents in a glomerulus, which is very predictive of end organ, renal failure. You have occlusion of the capillary lumen that also causes some ischemia. Podocyte injury, which is also very bad because podocytes have very limited ability to regenerate. You then result in tubular interstitial inflammation and damage, chronic kidney disease with systemic manifestations. So there are basically two models of immune complex mediated diseases in the mice that have been exploited by many investigators in the field. That the antiglomerular basement membrane nephritis, it's best actually to talk about nephrotoxic nephritis because there are additional antibodies besides just the glomerular basement membrane. But in any case, you have, when you take a naive mouse, you now give it a rabbit or sheep anti-GBM serum, you now see nice glomerular staining for the IgG, and within 14 to 21 days, the mice now develop huge leukocyte influx with neutrophils, macrophages, and T cells, hypercellularity, crescent formation that I just mentioned, massive proteinuria, and mortality in some cases. Now, this is what you would call the in situ. So this is a fixed antigen, which is a glomerular basement membrane to which antibody is generated. Here in spontaneous lupus, this has more a flavor of soluble immune complexes. Lupus-prone strains of mice exhibit a breakage of tolerance. You now get immune complex deposition. You'll see that this kind of deposition is very different in that it's sort of punctate in terms of, instead of the linear staining you see here with anti-GBM. Within 3 to 12 months, you also get a lot of similar responses of hypercellularity, neutrophils, macrophages, T cell influx, crescent formation, and proteinuria. Now, one of the very proximal events, of course, in inflammation in the glomerulus and elsewhere is the recruitment of leukocytes, which was of particular interest to my laboratory. The classical paradigm of leukocyte recruitment is the activation of the endothelium, then that leads to the leukocyte recruitment through the interactions of ligands expressed on the activated endothelial cells, such as selectins, with their counter receptors on the neutrophil. That leads then to firm adhesion and transmigration. And these, as I mentioned, selectins and integrins and ICAM and VCAM on the endothelial cells have been classically associated with leukocyte recruitment. However, when you've got immune complex deposition, we argued that there's probably additional receptors that are important in the actual leukocyte recruitment cascade. We hypothesized that Fc gamma receptors that are present on the leukocyte will directly be able to lead to leukocyte recruitment in the context of immune complex deposition. So now Take Tsuboi in the lab, so before I go on, if Jeffrey Ravage's group had shown that if you knock out all Fc gamma receptors, that means if you use a gamma chain deficient mouse, these mice are protected in various models of immune complex mediated disease, both in the kidney, they're protected from arthritis, glomerulonephritis, and many other immune complex mediated reactions. What we did here is to actually express a uniquely human Fc gamma receptor selectively on neutrophils and then cross those animals to gamma chain deficient mice to see whether now we were able to understand better what is the role of the Fc gamma receptor 2A on the neutrophil in neutrophil recruitment and disease pathogenesis. So these sets of mice, the 2A transgenic on the gamma chain background, as well as the gamma chain knockout mice, were then subjected to anti-GBM nephritis. And as you can see here in the gamma chain knockout animals, these are the glomeruli you can see here in low power. And on higher power, everything looks pretty much pristine, pretty much untouched, and no neutrophil accumulation, just as Jeffrey Ravage's group had shown. However, when we express the 2A on the neutrophils alone, and this is not an overexpression system, this is physiological levels of 2A, we found that we had great tubular atrophy, and in the glomerulus, you saw the sort of occlusion of the capillary lumen and adhesion of the Bowman's capsule, and this was associated with significant neutrophil accumulation, suggesting to us that the Fc gamma 2A expression on neutrophils, selectively on neutrophils, can give you neutrophil recruitment and damage. We wanted to know more directly how the Fc gamma receptor may be actually allowing neutrophil accumulation. So we made preformed immune complexes in vitro, gave them intravenously, and within an hour, when you surgically exteriorize the cremaster, Tracy Stockel in my lab had shown many years ago that leads to immune complex deposition in the postcapillary venules due to changes in the permeability of those vessels. So you can see here nice immune complex deposition, and we then now look by intravital microscopy, both in the gamma chain deficient mice, and this is a movie, and you can see there is some rolling, and that's due to, because the exteriorization will cause the expression of P-selectin on endothelial cells, and you get pretty fast rollers there. Not much adhesion or transmigration. However, if you now have a mouse that is expressing the human Fc gamma receptor on the gamma chain background, I think you can all see that it's, well, there's a lot of slow rolling. There's rolling, but there's a lot of slow rolling. And let me see if I can, oh, it's not buffering. Okay. There's a lot of slow rolling as well as adhesion and transmigration. So many people in the field have used knockout mouse models. In these two models of disease, anti-GBM and lupus nephritis, and as you can see, many of them, these inflammatory molecules, whether it be surface receptors, complement adhesion molecules, are pathogenic in nephrotoxic nephritis, as well as spontaneous lupus nephritis. I think what's missing here is the ability to know on what cell types these receptors are important and how they may be actually influencing the leukocyte recruitment, which will require the generation of conditional knockout mice. It will also require the ability to do intravital microscopy directly in the kidney, in the glomerulus, because as is right now, as I showed you, I did in the cremaster, because that's very transparent and easy to analyze tissue. But if we really want to know leukocyte recruitment in the microvasculature, we need to do some intravital microscopy. This is very challenging because the glomerulus is about, the closest glomeruli are actually about 100 microns from the cortical surface, and it's in a very optically dense tissue. So using two-photon microscopy, others in the field, such as Fred Geisman's group in London, has shown that patrolling, and these are fluorophore-labeled monocytes, GFP-labeled monocytes, increase their dwell time in the vascular in response to chemokines generated by a local TLR7 ligand within 3 hours of applying the stimulus. And then Michael Hickey's group also last year from Australia suggested an increase in neutrophil retention and neutrophil-generated oxidants within the glomerulus occurs within 2 hours of injection of an anti-GBM antibody. So this work has certainly paved the way for us to start looking very critically at what are some of the molecular mechanisms of recruitment in the glomerulus. Now, we were interested really in end organ damage and leukocyte recruitment and some of the mechanisms for that. So instead of using the lupus-prone strains of mice, which has loss of tolerance and everything else, but then it takes a long time to develop, we decided to generate a model of passive transfer of human SLE serum in what we call genetically susceptible mice, and we showed that induces organ damage and a role for neutrophils. And I'll just show you 3 slides on this because this work was published. But for reasons that I won't go into, we hypothesized that if we express the human receptors, but then remove a receptor called MAC1 from the mice, we now may make this mouse susceptible to developing disease. And you can see here we introduced human SLE serum and then collected the urine and serum over several days. And you can see that the renal function is significantly increased in mice that are lacking the MAC1, but expressing the human receptors. Those are called non-susceptible and susceptible mice. And the susceptible animals had a lot of endocapillary proliferation as well as crescent formation, as is shown in this histological score. And then one point that I just want to make here is that the human IgG immune complex and complement deposition was very similar in the non-susceptible and susceptible animals, and yet only the susceptible animals that lacked MAC1 had neutrophil accumulation, etc., some of the pathogenesis. So I think this is a very important and interesting point, and that I'll talk about in what we have the gap in knowledge right now. So we think that there's pathway 3 or something, some elements that are important. So you can have anti-nuclear autoantibody deposition, autoimmune response, but really there's something in the end organ susceptibility factors that allow neutrophil influx. So lessons learned, I think we have identified molecular and cellular mediators, renal injury, foreign glomerular immune complex deposition, and intravital microscopy recently will allow us to get further in that area. There's a lot of gaps in knowledge, and I won't have time to go into it, but I think some of the important points are, what are the target organ susceptibility factors that dictate the extent of the immune response and damage incited by the deposited antibodies? And I'll go down a little bit here, use multiphoton intravital microscopy in the glomeruli to look at the temporal sequence of events in leukocyte-induced inflammation. And then some interesting points is, how does the glomerular inflammation lead to tubular interstitial damage, which is most predictive of progression to end-stage renal disease? And how does glomerular inflammation actually affect increased risk for atherosclerosis, which is a macrovascular disease, independent of traditional cardiovascular risk factors? Thank you.
Open to questions. We have time for a couple of questions.
Malik, Chicago. Tanya, very nice talk. So, one of the factors in permeability across the glomerular apparatus is the charge.
Yes.
So the charge of the matrix and charge is really critical. So how do you think charge modification regulates this process?
So that's very interesting, because the glomerulus is actually very anionic. And so if you're cationic and you're small molecular weight, you're able to go across the filtration barrier. But there are some diseases where you may expect that the glomerular basement membrane charge may actually change. It's very rich in heparan sulfates, for example, in the glomerulus, and those changes may occur during diseases, which may also allow more deposition of extrinsic, trapping of extrinsic antigens.
Hi, Jose Lopez from Seattle. Do you think that there might be a role in humans for the FC receptor gamma R2A and platelets, which the mice don't have?
Yes, I absolutely agree. Definitely, there's a role for that on platelets. And as you just mentioned, since mice don't have that, it's very difficult to evaluate. We had a neutrophil selective promoter that we used. So in our case, we did not have any 2A on the platelets. Yes.
Hi,
Very
great
quick
talk.
question. Okay.
So, in your models, did you have a chance to look where the neutrophil extravasation actually occurs? Does it happen on the fenestrated areas or away from those?
We don't know yet, but hopefully we're going to get to it once we start doing some intravital. Yes.
Thank you very much, Tanya, and thank you, Esser.